Leicester. And I'm going to be chairing the session this afternoon. So there's just one little change to the programme in that we're going to run straight into the patient panel, which was due to take place after the tea break. And the tea break will then take place after the patient panel. Okay, so it's my pleasure to introduce the first speaker this afternoon, Dr. James Tomlinson. James is a consultant nephrologist at Imperial College NHS Trust, and he's going to tell us about his work in the field of transplantation. James.
Good afternoon. So pleasure to be here, and hopefully you can stay awake in your soporific mood just after lunch. But so it was just 20 plus minutes just going through what the relevance of IgA nephropathy is in kidney transplantation. So have I got? So just a quick overview how I'll cover it in 3 parts really. So why transplantation? Why do we need to think about it, and when? What does transplantation involve? And what does transplantation mean specifically for people with IgA nephropathy? And so I think probably by the fact that you're here, I think you grasp the basics of what the kidney function does for functions for your body. Now, it's multifold and varied. Obviously, the kidneys produce urine. It can control the fluids we get rid of out of our bodies, the excess fluids, and also the excess metabolic waste and toxins. By doing that, it very tightly controls the level of acidity in our body in different compartments, and also looks after the bone health by controlling various hormones that control your bone turnover, the bone strength, and obviously and how strong they are, and obviously reparation of bone as well. And then finally, many of you will know that the kidney produces a hormone called erythropoietin, which is something often abused by athletes, especially the cyclists, but something that's been transformed renal medicine in the last 20-odd years because it enables giving that EPO in kidney failure, enables us to treat the anemia. Now, so when the kidneys fail, you can see quickly why certain events happen. So we have problems with fluid balance. We often get swelling that collects in the ankles and also the lungs. We feel unwell, non-specifically unwell often, with itching, a bit of malaise and a bit of lethargy, and lack of sleep or inability to sleep with a lot of the toxins and waste that build up. Our bones get weak, the turnover is abnormal, and we have an increased risk of fractures. And we've become very anemic, so this is just showing the paleness or pallor that we can see in the mucous membranes around the eye when anemia is there. Now, fortunately, albeit there's about 10% of the population thereabouts have chronic kidney disease, but a very small percentage of those patients would progress to end-stage renal disease. In IgA nephropathy, the general rule of thumb is seen as since the time of diagnosis, about 1 in 10 people may progress to end-stage renal disease in 10 years. And an easy-to-remember rule of thumb is about 20% at 20 years. So they're the general ballpark figures of progression of renal disease related to IgA nephropathy. This is using UK renal registry data. In the table on the left, you can see that the biggest cause of end-stage renal disease across the board is diabetes. And in America, that's approaching, well, it's well in excess of 50% now. Now, below that is glomerulonephritis, of which only a small proportion of that is IgA nephropathy. So what are the options? If you or if one is going progressively advancing kidney disease, going towards end-stage renal disease, what are the options? And so we start thinking about things like dialysis. And there are different ways of performing dialysis. One is hemodialysis, where your blood is washed of toxins and the excess fluid is taken out. And that's a treatment that usually is 3 times a week for about 4 hours in a centre that can perform hemodialysis. And some patients can learn to do that at home, provided they have the motivation and the space at home to do that, then that can be performed at home. Equally, peritoneal dialysis is another form of dialysis that can also be done at home, where fluid is put into the abdominal cavity, and the lining of your peritoneum can act as a membrane to allow that exchange of fluid and toxins. Some people, often the more frail, sometimes more elderly patients, would choose what we call conservative care. So as with advancing kidney disease, you can get some symptoms that can be controlled with various medicines, and they would choose not to dialyse for various reasons. And that is an option, and often it can allow a better quality of life in their advancing years. But by far and away, the best approach for kidney failure, for end-stage renal disease, and to treat that to allow better outcomes is transplantation. I'm afraid the figure hasn't really come up well there, but that's supposed to depict a kidney being donated into the recipient, obviously. So just to recap, so what are the advantages of renal transplantation over everything else? Now, you can imagine dialysis is a very artificial and superficial form of renal replacement therapy, as we call it. So going intermittently into a dialysis unit or having intermittent dialysis is nowhere near as having a natural, normal kidney functioning there, second to second, minute to minute. So you can imagine that the overall control of all the fluids and the toxic waste is a lot better. The bone health is dramatically improved after transplantation, as is the anemia, and rarely you need to continue on EPO after transplantation. One of the very few graphs I've put into this talk is, I think it's quite descriptive in many ways. This is a big observational study from the US of A, showing the overall benefit. This is not IgA specific, this is just generally the transplantation benefit overall. So what they did is they had two groups. One, those that were transplanted, and two, over 46,000 people on dialysis but on the transplant waiting list, so effectively fit enough to receive a transplant. And you can see this dotted line across here, and this is above the line is an increased risk of death, and below the line is a decreased risk of death. So over time, on the bottom axis here, you can see at the start, you have close to threefold risk of death, and that's largely attributed to its major surgery, anaesthetic risk, and the complications therein. But that dramatically falls down over a short space of time, well within 3 months. The risk is dramatically reduced, and actually, in the longer-term benefit, it becomes quite clear that 50 to 80% increased or improved mortality, or improved survival, shall I say, with transplantation against those people that remain on dialysis. And how do we assess people for transplantation? So there are various criteria. Age is not really one of them. So yes, of course, we pay attention to age, but it's not really a barrier to transplantation. It's really depending on the person, the individual, and how fit they are. Gender, ethnicity are certainly not an issue in terms of being assessed for transplantation, but the important considerations are you fit enough to undergo a major operation and go through the recovery period afterwards? Is the person able to manage living with the transplant themselves? Are they fit enough to get themselves to the appointments? Can they take their medicines? Can they have the capacity to understand why it's important to take their medicines? Other medical conditions, obviously very important. How strong is your heart? Do your lungs work well? And are you very, very vulnerable to infections? And have you had any cancer? So all of these are taken into careful consideration. And there are rarely absolutes. It's really down to the individual about whether they would be potentially a good candidate for transplantation. And the workup period is anything between 3 and 6 months. So once it's decided that transplantation would be advisable and feasible, then we can work through the process. It should be well within 6 months. Now, how to get a kidney? There are two ways of getting a kidney, or two ways of donating a kidney. So one is a live donor, which is the preferred option because that can be a planned procedure. And that can be a live related, so that's obviously from a family member. Live unrelated is perfectly possible and preferable overall, either from a friend or a partner. And there's an increasing uptake of altruism in terms of people that decide they want to donate a kidney. They're not in close contact with someone that they know who needs a kidney, but they're more than happy to put it into the pool. And that is obviously coordinated through the same service to match the most appropriate person in the country on the waiting list. Clearly, there's very rigorous testing to make sure the donor is effectively undiseased, they're so-called normal. So we need to be very careful that we're not going to put them at a great disadvantage from it by putting them through an operation and living with one kidney. There are lots of advantages to this process against deceased donor transplantation, and that's largely because it's planned. So it's in a timely fashion. There's lots of pre-planning. You can eke out what the possible considerations are for those individuals before surgery. And there's no question there's better long-term survival in kidneys that are donated in this way. The second is deceased donor transplantation, whereby organs are harvested from a person who's met an untimely and often from, for example, a road traffic accident or having a stroke. And potential recipients are listed by NHS Blood and Transplant Service, and they're the coordinating body who would hold the register and also perform the matching and look after the logistics of making it happen on the day. Obviously, the consent of the family of the deceased is sought, and the kidneys are therefore matched to someone on the matching list in the country, so the kidneys are transported across the country. And so the average waiting time on that waiting list is about 2.5 years. It's based on many factors, but some of the more prominent factors are waiting time, your blood group, your tissue type, so what kind of proteins are on your blood cells and your tissue and your other cells that defines whether you'd be a good match for a kidney or not. And clearly age is important. So for instance, if a kidney is donated from a 70-year-old person, it's not ideal to put that into a 20-year-old recipient because you expect a longer life expectancy in the recipient. So you want to have them as age matched as possible. But obviously that's down to the individual case as well. And what we find, certainly in West London, as in many inner city areas, there's a mixed population. So a lot of our potential transplant recipients are from other countries, which, if you think about it, if our donor pool is largely, still in our country, is largely Anglo-Saxon or British Caucasians, that means that the people dying would have a very different tissue type to the people potentially wanting a kidney in some parts of London especially, but obviously in other inner city populations. So that can limit your chances of getting a transplant in X amount of time. It can lengthen the time you're waiting on the list. So the actual transplant operation, I know some people have been through the process and know this all too very well, but certainly in IgA nephropathy, your kidneys wouldn't be taken out ordinarily. Your own kidneys would remain in, and you receive usually one kidney. Some centres are doing 2 kidney transplantations. That's often when there's a size mismatch. So if there's a paediatric donor into an adult, sometimes 2 kidneys would go in. But by and large, it's 1 kidney that you get in addition to your failing or failed kidneys which stay in. It's put into the pelvis and attached to the artery, to the vein, and to the ureter, which is the pipe leading from the kidney to the ureter plugs into the bladder. The mean time of the admission is about a week, give or take a couple of days, depending on progress. And the thing to note is clearly in clinic you'll be seen every 2 or 3 times a week with quite intensive follow-up to look at, well, overall how you're doing. Are you drinking the right amount? Are you coping with the pain and the wound healing, and what the blood test is showing in terms of kidney function. And you've monitored extremely closely because there is quite a bumpy phase, and the kidney function can change according to how much you're drinking and various other factors. So we monitor that very closely. And then towards the 3-month mark is when things tend to settle, and then I think the longest we'd wait before seeing you again is every 3 to 4 months in clinic when things are all well and good. Many people are returned to work by 3 months, but it's a big process. It's not to be underestimated, but overall the longer-term gain is very much greater than remaining on dialysis, so it's certainly a worthwhile investment for many people. Importantly, it's, after you're transplanted, went to your home, that is the time when people really have to take control of their own care. So looking at medicines is one of the key aspects, really, and understanding what the medicines are and understanding what the side effect profiles are and how to manage those. So this really splits up the types of medications into 4 different groups, really. So the key thing is the anti-rejection. So these are the immunosuppressants that are used to dampen down your immune system to prevent your immune system attacking the kidney that's effectively a foreign body. So the prednisolone, the steroids, things like tacrolimus and mycophenolate can all be used and are very helpful to dampen down the immune system. Blood vessel health, as ever, in any kind of kidney disease is hugely important. So there's lots of strict attention to controlling the blood pressure, using blood thinners to avoid clotting, in especially the small vessels, and controlling your lipids and your cholesterol. Certainly, if you're diabetic, then optimal control of that is always preferable. and you'd be put on lots of medications, including things to dampen down the stomach acid and prevent indigestion. Because we're dampening down your immune system, we do use agents to help prevent infections with viruses and also with bacteria, and often, certainly in the initial phases after the transplant, would give you those kind of agents to reduce the chances of infection. So there's a lot going on, but clearly throughout the whole process, people are counselled very well, and we have a strong presence of pharmacists in each clinic often as well to help people through that. So this is in general again, so not necessarily specific to IgA nephropathy. How long does the transplant last? So some can last well in excess of 30 years. There's clearly a huge spectrum from suddenly immediate failure from perhaps a surgical complication, very rarely fortunately, all the way up to 30, 40, 50 years. Now, those causes of failure are obviously large and wide from the surgical complications, but some form of rejection, either acutely or over time, is always a greater risk. So that's why one of the reasons why we monitor it very closely in clinic. Clearly relevant to this information day is IgA disease, and we'll talk about that in the next couple of slides. And sadly, some people's grafts that do fail do require going back to dialysis or starting dialysis. But certainly retransplantation is completely possible and is done clearly with lots of pre-planning. But we're looking at around an 80 to 90% good kidney function or achievable kidney function within 5 years, but sadly over time that does drop off as the natural reduction in graft function. Now, specific to IgA disease, I think there's important things to consider in a population with IgA as opposed to a mixed bag of, say, diabetic nephropathy or any other causes. So people with IgA entering the realm of having to have a transplant tend to be younger, and they tend to be healthier because they don't often have coexistent diabetes and various other conditions that have led them towards end-stage renal disease. So a lot of this is on observational studies, largely US, but there's a couple of very good Australian and New Zealand registry data producing a lot of informative numbers in terms of IgA in transplantation. So certainly people with IgA tend to have an increased, they're more likely to have a live donor transplant. Certainly the wait list time, if they're on the waiting list, the wait list time is shorter. And one of the US studies suggested that if you're on dialysis with IgA waiting for a kidney, you're almost at 15 times greater in the first year of getting a transplant than someone who hasn't got IgA nephropathy. So once the kidney's been transplanted, are you likely to get IgA coming back in the new kidney? And the bottom line answer is yes, it's fairly common, but there's a whole spectrum of how severe and what that disease really manifests in terms of severity. So what we measure in clinic for anybody, and especially in IgA, clearly looking at the blood tests according to serum creatinine, which is a surrogate marker of kidney function, urine dipsticks and measuring protein, how much protein in the urine, and also fundamentally, if we're concerned about any change in this, then we'd be looking at kidney biopsy, so getting a small piece of kidney to identify what's happening to the cells in the kidney themselves. And that's really obviously the gold standard to define any kind of disease in the kidney, and specifically with IgA recurrence. And so what many studies have found a varying range of recurrence in grafts, but when they're looking at the biopsy cohorts, they see about a recurrence in about 60%. Now, many of those actually haven't got any significant disease. The blood function, the blood creatinine according to function doesn't change. Some of them don't even have any dipstick positivity. So there's a whole spectrum of recurrence that go to from subclinical, i.e. not detectable in any other way, to quite aggressive disease. And so while that number 60% is alarming, I think we have to understand the context. So those people at high risk of recurrence in the kidney transplant are those with their primary disease and their native kidneys being more aggressive and more abrupt. But interestingly, as ever, it's a complex disease. So in the same patients, and these are obviously small numbers in some studies, but in some patients that are repeatedly transplanted on their second or third kidneys, they don't necessarily run the same course. So I think if otherwise feasible, it's still worthwhile considering retransplantation, even despite losing the first graft to IgA nephropathy. Interestingly, I think Daniel Gale's talk coming up will go through the genetics of IgA nephropathy, but there is clearly a signal in a lot of the observational studies where we see those people at higher risk are also those people receiving a live related donor, and if the matching is especially good, if it's especially close, interestingly, you have a chance of recurrence. Some people have suggested that actually monitoring IgA levels post-transplantation, if those are especially high, you do have a higher risk against those people that have undetectable or normal range IgA in their bloodstream. And others, there's been one large study and several others to support this idea that actually patients being brought off steroids entirely run a higher risk of having IgA recurring in their graft. Depending on the centre, some transplant centres will keep you on a low dose of prednisolone long term, and some will try and get you off that, and it depends on the local protocols. However, those that are taken off steroid look like they have a slightly higher risk of recurrence. And so, if it does recur, what can you do? So, there is little evidence to suggest or to guide doctors and people with IgA about what best to do in terms of changing the medicines. So, all the immunosuppression that has been tried in native IgA disease has also been tried in transplantation and recurrence, with varying success. So there's always case reports of great success, but in bigger studies, the signal isn't necessarily straightforward. People have also tried tonsillectomy pre-transplantation to reduce the risk, and that seems to have no great effect. And the steroid effect, as I've said before, there seems to be some indication that's a real effect. As always, ACE inhibitors, so the blood pressure medications that are good for blood pressure but seem to have an anti-inflammatory effect as well, they're also shown to be useful in the progression of IgA in the graft. But overall, I think it depends on the individual case and how aggressive the disease is about what may be chosen as an immunosuppression to try and treat that reoccurrence. And an important thing is, across all the observational studies, the outcomes are not that dissimilar in transplantation compared to people with non-IgA disease. And I'll just show you this slide. So I'll talk you through these graphs. What we have is this is an Italian study of 200 patients with IgA nephropathy being transplanted, and the comparator group was about 400 patients without IgA nephropathy. So that was a mixed bag of diabetes and various other diseases. The dotted line is the patients with the IgA nephropathy. So you see at the top left in graph A, this is patient survival. So this is the survival of the person themselves. And there's no significant difference. You can see a separation, but that's not significant. So there seems to be no obvious effect on long-term patient survival between the groups. If you look on the bottom left at B, you can see this is now graft survival. So this is survival of the kidney. And there starts to be a separation between those people with IgA and those people with non-IgA disease, in that over time, especially over 15 years, there starts to be a separation whereby those people with IgA nephropathy are more likely to lose the function in their graft. Another interesting finding in the study actually was there seems to be a changing incidence of IgA recurrence in transplants over time. So if you were transplanted in the 1980s and then the 1990s and in the year 2000s, you have a decreased risk of reoccurrence. So what they're finding is that, at least in this study, that their recurrence in the graft of IgA was going down over time. And that suggests it's probably related to the changing protocols of immunosuppression at the start of transplantation and then the maintenance. And so it's very difficult in a relatively small study to unpick exactly which agent may be better, but there is certainly a suggestion there. So there clearly is a response to different regimes of immunosuppression with regards to recurrence. So I'll finish off with some take-home messages, really. End-stage kidney disease can occur, and it does occur, fortunately, in a minority of people with IgA nephropathy. Transplantation, by and large, is the treatment of choice for longevity and quality of life. And there are huge benefits, and this is across the board in kidney disease, about planning early. So, as always, a preemptive transplantation is so much better than going through transplantation after having to need to start dialysis. So that word preemptive really means before the need to start dialysis. And remember, there are always options. So I think along every step of the way. So obviously be sure to discuss it with the teams, nephrologists and all the specialist nurses and anyone who's involved in your care. Okay, I'll finish there.
I think in order to stick to time, I think we just time for one question.
So we've got a question from people watching at home. They ask, can people with IgA and people who've had kidney transplants drink alcohol?
Yes.
Yes.
But so as not to fall over, I think. But no, I think the general advice would be the same. So yes, always in moderation and under the recommended limit, which I forget the numbers for these days.
One more? Yeah, okay, one more and then we can.
And someone else has asked, what happens if you react to tablets? If you can't take tablets specifically, are there other ways you can take the drugs that you need?
As always, with any medicine, I think it's important to understand exactly why people can't take tablets. Are the tablets too big? Is it a more... Sometimes people have trouble swallowing anything foreign that's not food. So I think it really depends on the scenario. But as I said on the last slide, there are always options. So I think just talk through what the issues are and whether it's down to a specific tablet, and then there are often alternatives and different even preparations of medicines as well.
Okay, thanks very much. Thank you. Our next speaker this afternoon is Dr. Daniel Gale. Danny is an associate professor and honorary consultant nephrologist at the Royal Free Hospital. He's going to explain to us how genetics can impact on IgA nephropathy.
So thank you for the opportunity to talk to you, and thanks for listening to me today. So I'm really going to give you some background about genetics, what we mean by genetics, and how that relates to IgA nephropathy. So genetics is about genes, and genes are the unit of instruction that our cells carry in order to make the proteins that are the building blocks for cells that give them the ability to do all the things that our cells need in order to come together and form a functioning organism like a person. Each cell in our body has got 2 copies of almost all of our genes, but only a proportion of those genes, those instructions, are switched on at any one time. And it's the particular genes that are switched on that determines which proteins are being made by a cell, and that determines whether the cell is making fingernail or in your eye or in your ear or in your brain or behaving the way it should. But the important thing is that all of our genes are present in almost all of our cells all the time. Genes are encoded in a chemical called DNA, the structure of which was discovered in the 1950s. And DNA is quite a special substance because it forms this rather beautiful double helix, which you may have heard of, and it comes in 4 different letters or bases. So these are different DNA molecules that stick together into a double helix, and they are made by bases called adenine, thymine, cytosine, and guanine, or A, T, C, and G. And it's the sequence of A's, T's, C's and G's in this molecule that is the genetic code that encodes all the genes. And it's just like a book in that each chapter is a gene. The chapter is made of a series of letters that come together to form a word, a sentence, and a chapter of the book. And these are genes joined together in very long molecules called chromosomes. And the observant among you will have seen that A's always pair with T's, C's with G's, and so this code, you have two strands of DNA that stick together, and that allows the cell to then replicate the DNA, and that's the sort of fundamental building block for life. So this is another picture that shows the DNA molecule with the sequence of, we call them bases or individual letters, and it's wound up really tightly on special proteins, and they all come together to form chromosomes, and we have 23 pairs of chromosomes in almost all of our cells. If you unwrapped the chromosomes in a single cell and put them, all the chromosomes end to end, you'd get 2 meters of DNA. So that's how tightly it's lined up. And because we've got around 35 trillion cells in our body, that means that if you put all the DNA from all of our cells together, it would stretch twice across the diameter of the solar system, which is too big a number for me to really imagine. But just to give you some idea of how tightly packed up this molecule is. Now, the genes are arranged, the name for the collection of genes in an individual is called the genome, for fairly obvious reasons. And the genome, which is the book of life, and there's a printed out version of the human genome upstairs in the next door building in the Wellcome Trust. You can actually see it from the street. And it contains 3 billion DNA bases. So we've got 2 copies of our 3 billion letter encyclopedia that is the instruction manual to make a human being. The way they're arranged, genes actually only cover a small proportion of the DNA sequence. There's big gaps between each genes, and those gaps probably play an important role in which genes are switched on at any one time. And the genes themselves have small parts that are eventually made in some substance called RNA and then copied into protein, and that's how the cells make the protein. The details are not really important, but what is important is that the non-coding regions of our genome occupy 98%, and the small chapters that actually tell us how to make the proteins that build us are only around 2%, and only a small proportion of them are known to be associated with any disease. So that's the basic structure of the genome and the genetic material that we all have in our cells. The genomes of any 2 people, apart from identical twins, are very similar to each other. Identical twins are almost 100% identical for obvious reasons, but for the rest of us, any pair will have between 99.6 and 99.9% identical sequences. And this differences, this very small proportion of difference, explains why we all look, sound, smell, act, behave slightly differently. It accounts for some of that variation. There's, of course, how you're brought up will affect other things as well, affect how you behave as well. The human genetic sequence is also 99% identical to that of a chimpanzee. So the amount of, most of the DNA that we've got in our bodies that makes proteins is shared with animals that are closely related to us, and the further across the tree of life you go, the more dissimilar animals are. But clearly most of the things that a chimpanzee has to do in order to stay alive is very similar to the things that we have to do to stay alive, and there are small differences there, obviously critically important in terms of how the animals look and behave and all the things that make us different from chimpanzees, but it's only encoded by a very small proportion of difference in our genetic sequence. So that 0.1 to 0.4% difference is at around 4 or 5 million particular places in the genome where letters tend to be different between individuals. And about 3 million of these are common variants, and we call these single nucleotide polymorphisms, or SNPs. And I'm going to use the word SNP a little bit later on in this talk, and I'm not talking about a political organisation north of the border. What I'm talking about is the normal variation between individuals. And SNPs, there are other sorts of variation as well, but SNPs are the most important ones, or the most numerous ones, and about up to 3% of them actually have a function. So for instance, the difference in eye colour that you'll see around the room is because of SNPs, so polymorphisms, single nucleotide polymorphisms, or SNPs that don't have are not dangerous in themselves, but just account for the differences between people. Many of the SNPs don't encode any difference at all. They're just differences in the letter, rather like a misprint in a book that doesn't change the sense of a word is not going to change the understanding of the reader. So that's the genome and the genetic material, how genomes differ from each other, and how does this manifest as in sort of everyday life? Well, most of us are quite familiar with the idea of genetic traits or genetic diseases, which is where a characteristic in one member of the family is then passed on through other members of the family. We sometimes call these monogenic traits or Mendelian traits after Gregor Mendel, who first described them in pea plants in the 19th century. So one copy of our genome, you'll remember that I mentioned we've got 2 copies of our genome in all of our cells. We pass 1 copy of our genome to each of our children, and the genome is shuffled up in that process, so it's a different half of our genome to each child that we have, and they get the other half from the other parent. So they have half of their genetic material in common with each parent, but which half it is not known before is not predictable. Some genetic variants, such as SNPs, can have a big effect on the function of a gene, and eye colour is a really good example of this because it's something that doesn't cause disease, but is very obvious. And there are 2 common variants. There are less common variants as well, but there's 2 common variants encoding brown and blue eyes. And if you have either 1 or 2 brown versions, and versions of variants we call alleles, but that just means version of a gene, you have brown eyes. If you've got 2 blue versions, you've got blue eyes. And that means that if 2 people who have 1 brown and 1 blue-eyed version of the particular gene come together and have children, they will each have brown eyes because they've got 1 brown version. That we call as dominant over the blue version. And the children might inherit 2 brown copies or 1 brown copy from one parent and a blue copy from the other, or vice versa, and they will all have brown eyes. But the 4th child, and of course it's random, you might have 10 children and none of them inherit both blue-eyed versions, might have blue eyes. And that's sort of the simplest model of genetic inheritance of single gene defects, and that's what we mean when we talk about genetic diseases. And I put that there because not everyone is clear about where the difference between a genetic disease is and where a disease that has a genetic contribution is. And I'm going to move on from single gene defects and talk about what genetics call complex trait genetics. And all this means is these are traits in which genes make a contribution, or genetic makeup makes a contribution, but it's not as simple as 1 gene, 1 disease, or 1 gene, 1 trait. It's the constellation of gene versions that you inherit that determines, say, your risk of disease. And we know from talking to patients and probably talking to each other that some diseases like heart disease, cancer, dementia do tend to occur in families. People will either have lots of people in their families being affected by these conditions or maybe not so many. And clearly there's a genetic contribution to it. But these diseases, by and large, are not directly inherited from a parent such that children have a high likelihood of getting the disease. It's just increased compared to the general population. And the biological process that's underlying this is the inheritance of multiple copies of multiple different genes that each carry a slightly different risk of the disease with them. And one example of such a trait is height. So we know that height is inherited because, in general, tall people have tall children and small people have smaller children. But if you look carefully, you'll see that very tall parents will tend to have tall children, but they won't tend to have very tall children on average. Likewise, professional athletes will tend to have sportingly talented children, but they won't tend to have professional athlete children. And the reason for that is to do with the multiple different genes that encode for each or that contribute to these sorts of traits. So taking the example of height, what I've got here is each of these circles represents a gene. It doesn't matter what the gene is. And the green copies are the ones that just boost your height by a very small amount, say an extra 2 centimeters. And if you carry, the more of these green ones you have, the more you're genetically programmed to have a greater height. And you'll see that some people have 2, some people have 3, some people have 1, and there will be slight differences in their height. And every so often a person will come along who's got 6 of these very tall genes, and they will be quite a lot taller than the general population. Now, when each of these people, let's say these 2 individuals, a man and a woman, say, have a child, they pass on half of these to each child, and this child, for instance, will inherit just 1, but they could equally have passed on the other set of genes that they've got, in which case the child might have inherited 2. And likewise, in this pairing, they will, this child, say, will inherit 3 alleles, so will be similar to this parent. But if this parent, who's got 6 alleles, passes on 3, it doesn't matter who they reproduce with, there's no way the child can get, or it's unlikely for the child to get a similarly large amount. And this is because this causes what we term regression towards the mean, which is that the risk in an individual, the genetic risk in an individual might be high, but with each child, it tends to get closer to the general population's risk. And that, I think, is important for height, but also important for diseases like IgA nephropathy, where it's not the case that you either do or don't inherit a gene that causes the disease. It's that you're born with a set of genes, some of which encode a somewhat increased risk. And scientists and clinicians have been looking a long time for single genes that cause IgA nephropathy, and none has been found. That doesn't mean that there definitely aren't any, but science is not aware of any single gene changes that directly cause IgA nephropathy. But what they have seen is that IgA nephropathy does crop up in families more often than you'd expect by chance. And there's very clear differences in the prevalence, the rate of IgA nephropathy in different countries. So in East Asia, for instance, the rates of IgA nephropathy are quite considerably higher than in Europe and America. And this risk also applies to people with East Asian ancestry who live in the UK. And that very clearly suggests that there's a genetic contribution to this. But it's not like eye colour. It's not something that you pass on directly to your children. It's a risk that in future generations tends to approximate the general population's risk. So why study the genetics of IgA nephropathy? The advantage of studying it is that it gives you some insight into what the underlying biology of the disease is. It's not a way of testing people or directly offering them a cure, because it's not telling a simple explanation of what causes the disease. Rather, it's a way of seeing which genes are important, therefore, which are the biological processes associated with those genes that are contributing to the disease. And in order to do these sorts of studies, you need lots of people. Lots of people with IgA nephropathy, for instance, and lots of people without IgA nephropathy. And then with modern genetic technology, one can sample a large number of different genes and see which alleles are of a higher frequency in people with the disease compared with without the disease. And the findings from this study, which I'll show you, these sorts of studies, which I'll show you in a minute, tell us that each particular variant contributes a small amount of risk. You know, we're talking 5% to 20% increased risk, depending on if you summate all the different genetic variants that people have. And very clearly we know that people with a high genetic risk may, for reasons of their environmental exposure or reasons we don't understand, never actually get the disease. And that some people who have a low genetic risk may still go on to get the disease. And that's just the fact that we're dealing with risk factors, we're not dealing with direct cause and effect. And this means that this sort of genetic information is really not terribly useful in an individual person. If you take a person with IgA nephropathy and find out whether they were at high or low genetic risk of the disease, it may be interesting for them on an academic level, but it doesn't take you forward in terms of making clinical decisions, and it doesn't really inform what the risk to their family is, which is slightly counterintuitive. But it is very useful if you want to understand the disease. So this is the result of the first Genome-Wide study that was done in the UK, published almost 10 years ago now, and this shows association at one place in the genome. So this graph is showing you every chromosome, and it's sampling about 300,000 different variants across the whole genome. So it's a very small sample of the 3 billion letters that we've got. But what it shows is that certain genes at this one place in the genome are associated with the disease. And that place is in the what we call the HLA locus, which is the part of the genome that one matches when one does a kidney transplant. So this is part of the immune system that tells people, that tells the body which proteins to attack and which not to attack. Subsequent larger studies have been done in China and the United States, and they've replicated this original finding and also identified other genes. The gene names are not terribly important at the moment, but what it's showing you is very clearly there are certain HLA types or tissue types that are associated with IgA nephropathy, and this probably provides at least some of the explanation for the increased recurrence risk in very closely matched kidney transplants. Subsequently, these genetic studies have all been put together to give a sort of catalogue of the genes, variants of which seem to confer a significant increased risk of IgA nephropathy. And if you look carefully at all of those different genes, almost all of them are known to be involved in the immune system. Some of them are involved in how our bodies make antibodies, but other of them are involved in how our blood kills microorganisms. Others of them are involved in how our surfaces defend themselves against infection. So it seems, just from looking at what the genes are, one can see that the genes that are important, that are different in people with IgA nephropathy, are genes that are important in fighting infection. And you can compare those genes with genetic associations in other diseases. Again, the details aren't important, but there's overlap between IgA nephropathy and some other autoimmune type diseases. Again, diseases where the body's immune system is slightly overactive in one way or another and causes damage to the body. And one can also look at the burden of these risk alleles, or the versions of the genes that are associated with disease, and see that they correlate really quite strongly with the diversity of infectious worms in an environment. So we're talking about pinworms, hookworms, that are not terribly common certainly in modern Britain, and weren't that common in pre-modern Britain, but were a lot more common, and importantly, a lot more different types of infectious worms were and are present in parts of the world where IgA nephropathy is common. This doesn't prove it, but it does suggest that our body's ability to respond to and destroy certain types of infections is what has driven the predisposition towards getting IgA nephropathy. So it's an upregulation of our immune system, and it seems to be associated or correlated with the types of infectious organisms there are. We can also use the genetic information in another way and make a genetic risk score. Now, I said before that adding up the genetic risk in an individual isn't terribly helpful, but if you do it in a cohort of people, then it can provide some important insights. I should say that there are so many risk alleles, versions of high risk that have been described, that almost all of us will have one or more of these risk alleles, whether or not we've got IgA nephropathy. But what we can do is compare the distribution of these risk alleles in different groups of people. And one way that this has been done is looking at the the frequency of these risk alleles across the world. And this fits with the infectious disease hypothesis in that the parts of the world where there's a great diversity of infectious parasites have a higher risk score for IgA nephropathy, and there's a lower risk in other parts of the world. It's not quite as simple as that, because the risk for IgA nephropathy is much lower in people of African ancestry, and there are likely to be good reasons for this, not all of which are understood. But we can also look at the genetic risk of IgA nephropathy in the general population. And there's a national study running at the moment called the UK Biobank, which recruited around 400,000 people from the general population, so they were unselected, and tested their genes looking for common variants. And we can look in that data and find what every individual in that study's genetic risk score for IgA nephropathy is. And what we can see is that the people in the study, the 400,000 who don't have IgA nephropathy, that's the distribution of their risk score. So it hovers sort of around the 3.9 level, and that's just an arbitrary number that's made by adding up the number of risk alleles in each person. And people with IgA nephropathy, the average risk score is somewhere between 4 and 5. So you can see that if you, as an individual, have a risk score of, say, 4.1, well, you could equally be from the general population or from the population of people with IgA nephropathy. It's not terribly helpful. But if you look at tens or even hundreds of thousands of people, you can see that these populations are different. And we can do that to look at people in UK Biobank. So 13,000 of them were known to have hematuria, or blood in the urine, which we know is one of the features of IgA nephropathy. There are other things that cause blood in the urine, and these people had only blood in the urine, and they'd never been undergone a kidney biopsy. So the cause of their blood in the urine wasn't known. And what you can see is that there's a very slight, so that's the healthy population without blood in the urine or kidney disease. So 320,000 people have a genetic risk score of 3.9. People with blood in the urine have a very slightly increased genetic risk for IgA nephropathy. And this genetic risk goes up if you've got protein in the urine and if you've got high blood pressure as well. And if you've got a diagnosis of IgA nephropathy, which is a much smaller number, then the genetic risk score is very much higher. And what this is telling us is that, well, one explanation for this observation is that 20% of the people in UK Biobank with blood in the urine, their increased genetic risk would be explained by them having IgA nephropathy that's not been diagnosed. And the implication of that is that IgA nephropathy is a lot more common than we generally recognise, and it's also a lot more benign in that there's this quite large, 20% of this 13,000 people out of a sample of 400,000, quite a large number of people who have blood in the urine, probably have IgA nephropathy. We don't know because we can't biopsy them, but have the genetic risk for IgA nephropathy, and that would be explained by them having mild IgA nephropathy that presumably will never do them any harm. So that's a whistle-stop tour of some of the things that genetics have taught us about IgA nephropathy, and I'm very happy to take any questions.
Just on your last slide, just clarify the number in the sample, the overall sample.
Yeah, so this was about 400,000 people in UK Biobank. 320,000 didn't have any evidence of kidney disease. 13,000 have just a little bit of blood in the urine. So it's quite common. About 1% of the general population are thought to have that as well.
What's the hypothesis with regards to the abnormal pattern of glycosylation on IgA and how that collects in genetic?
So I didn't have time to talk about that, but we've done some work in the last few years that shows that there is a very clear genetic driver for IgA glycosylation. In other words, there's variation at certainly one gene and probably a second gene that controls the sticking on of that glycosylation group onto the IgA. And differences, the genetic difference between people in one particular gene seems to explain the very strong heritability, which is the inheritance of abnormal IgA glycosylation. That signal doesn't come through as a genetic risk factor for IgA nephropathy, and that's because the studies that I showed you before had up to 3,000 or 4,000 people in them, and you'd need studies of 20,000 people in order to test the hypothesis directly that genetic predisposition to galactose-deficient IgA is a risk factor for the disease. And that's work that's going on at the moment and might be able to give us one day give us a really good answer to your question, which is what is the causal role for galactose-deficient IgA in the disease?
Hello, this is something that you have been speaking about in the talk, but it's something that our viewers are really interested in, so I thought it was worth going back to. So does IgA run in families? Are people likely to pass it on to their children? That's what I want to know.
So it's a really good question. It's very clear that familial clustering of IgA nephropathy is well recognised. So if you look at a population of people, then there will be certain families that crop up in which multiple people in the same family have got the disease. Now, this has been well described in medical literature going back at least 25 years and probably more than that. And these families have been very extensively investigated with genetic techniques which have moved on a lot over that period of time. And we haven't yet found a really clear causal gene. So it's not like eye colour. It's that family has a high background risk of IgA nephropathy, and that risk may manifest itself. But it doesn't mean, and with each generation, the risk one presumes will be getting less, provided the family is not in a geographical isolate, in which case it's a little bit more complicated. So yes, IgA nephropathy can occur in multiple family members, and it's well recognised, and it probably reflects families with just a slightly higher genetic risk of IgA nephropathy, but it's not really been proven to be a directly inherited trait like eye colour, where somebody whose parents got IgA nephropathy has, say, a 20 or 50% chance of the disease. That's never been shown.
Are you looking at the impact of epigenetics on IgA and looking at the influence of sort of the non-coding parts of the DNA and how they are controlling sort of gene expression? With the link between sort of what was the thing about the worms and things and sort of the environmental impacts on why more people in Southeast Asia have IgA compared to over here?
So that's really two questions, and I'm going to answer your question about the non-coding regions of DNA, and to tell you that almost all the genetic variants that have been associated with IgA nephropathy are in the non-coding regions of the DNA. And I think that's something that's come out of other similar studies in other diseases, that it's not the actual gene that's different in people with the high risk compared to the low risk. It's the amount of the gene that's made. And the part of the genome that controls or that has an important influence on how much of any particular gene is made is the non-coding regions near to that gene. And we are sort of at a very early stage scientifically in being able to understand how changes in the sequence of the non-coding parts of the genome affect gene expression or other aspects of gene regulation. So, for instance, the galactose-deficient IgA, the IgA glycosylation differences are very clearly coming from a non-coding region that's near to this one gene. And differences in that one region, that's work that we're doing in my lab at the Royal Free at the moment, are very clearly have an effect on the amount of that protein that gene that's made by the cell. So yeah, non-coding sequence is absolutely critical to getting the balance of gene expression right in the cell. Epigenetics is much more, it's quite a different scientific field really, because again, that's a mechanism for control of gene expression, but it's not fixed in an individual for their whole life, and therefore it's not always clear that what's cause and what's effect. So it's a much more difficult problem to solve, and probably the stronger information that we've got at the moment comes from genetics, and our ability to understand epigenetics is quite rudimentary at the moment.
Okay, thank you. In the interest of time, I think we need to press on. Okay, thank you very much. So the last speaker of this session is Professor Frederic Tam, who is a consultant nephrologist at Imperial College. Fred is going to give us an update on his clinical trial testing the drug Fostamatinib in IgA patients. Thank you.
Thank you for the invitation today. I think I see quite a number of familiar faces around. Some of them are the patients or collaborators I met over a previous meeting. And some of them are the patients I see in the clinic as well. So it's wonderful opportunity to share with you. Now, see if we can put on the slides there. Yeah, this one, yeah. Okay. Great. Yeah. Great. Okay, so we're slides now. So I've been involved in quite a few of these meetings over the last few years. It's quite exciting to give you an update of what we have been doing. Now, so what I'm going to summarize to you is that some of the research work we've done with a lot of collaboration with the patients and also different hospitals, both in UK and around the world. And the main thing to all of you already know the importance of kidney disease. The main story I want to bring to inflammations, because this is the center focus of IgA nephropathy. Now, I've been working in a few miles away from here for many years. On one of the rare days, I managed to capture the blue sky there. And the most important thing to show you here is that this is the main entrance of our hospital, Hammersmith Hospital. What I try not to picture is the one which grew up a few yards away from there. And my boss always tells me if I do anything wrong, I'll go the other way. And this is our renal unit. This is a clinical center formed by amalgamation of 3 NHS centers about 13, 14 years ago, St. Mary, Hammersmith, and Charing Cross. And this is important because this is what our basis of all our research work is all focused on patients. And this is our laboratory. The good news is that all these are within 5 minutes walk from each other. Because a lot of the challenge of kidney research is that the hospital is so remote away from the research center, you can never link things up in a meaningful way. And one of the concerns in IgA nephropathy is that we know IgA is contributing to inflammation and scarring, resulting in kidney failure. And if hypertension is not being controlled, it makes the whole thing worse. But other than that, we actually know very little about step-by-step deterioration of the kidney. And one of the problems is that early, as you, Jonathan, may have mentioned early on to you today, early stage of disease is not always clear-cut. And then, although we got lots of therapy around in kidney medicine over the years, and some of you may have come to a kidney clinic, you know that there are medications around. So what's the problem? The problem is that some of the medications are very generic, so they're being used to treat a wide range of conditions. Sometimes they work, sometimes they don't. And even when they work, if they're more generic or immunosuppressive therapy, they tend to increase the risk of infection and other complications like diabetes. So what we think of is that we need to do something a little bit more. Hopefully we develop something more effective in a reliable way and hopefully less side effect. But of course, all these are our intention. The question is it true or not? I think that's where research comes in. Now, so all of you are aware that how it's been diagnosed in the screening stage about urine dipstick test, but the most important thing is kidney biopsy. The reason I bring up the kidney biopsy here is that these green color, they are the antibody IgA got stuck to the kidney of patients, causing inflammation and fibrosis. And this is why IgA nephropathy is being named as such about 50 years ago. And the question is, what happened after that? Now, this is where our limitation understanding is. Now, as a researcher, as a clinician, all of us only know a tiny bit of the whole story. And we are quite blind to what the overall picture is. And my personal understanding is that a lot of my research have been to how inflammatory cell would penetrate the kidney and causing tissue damage. And just bear in mind that don't believe anything I tell you superficially, because you'll hear lots of researchers like Danny, who's interested in genetics. They have a different angle of how to understand the disease. And all these different angle understanding is essential, because if we sort of being blinker and one explanation of everything, like my bias of inflammation, we would never get the full understanding of that. Now, so one of the molecules I've been working on is spleen tyrosine kinase. Now, some of people ask me, why do you work on this molecule? When inflammation is such a big story, you can't say one molecule explain everything. And that is absolutely right. And I think when at the end of the story, I tell you, just bear in mind that there are many other targets we need to think about in the bigger picture, rather than focus on one molecule. And the second thing is that, actually, regretfully, when I look at some of my older paperwork, my answer is, why didn't I study it earlier? Because 2 years before I started this project, I was having open collaboration discussion with a lot of other scientists. There are a couple of people telling me, write this down on your piece of paper, spleen tyrosine kinase, this is important. And what did I do to a piece of paper? I put that away. And it's only many years later I discovered that piece of paper was there. And by that time, I eventually have started working on this molecule. Now, the importance of this molecule is that the honest truth is that we don't know whether it's important in IgA nephropathy. What people know is that spleen tyrosine kinase is an important enzyme in our body. All of us got it. This is important in our defense system. That is what helps our lymphocyte to make antibody response to bacteria and viruses. That is involved in controlling inflammation, how we fight pathogens. So there are several functions that are known to be in our immunological system. But what has not been known is whether it's anything to do with IgA nephropathy. And why didn't we know about that? Partly is a typical scientist is that I'm scared to find out why it's involved or not. I remember a few years ago, or probably getting near 6 or 7 years ago, one of the visiting researchers in my lab, and she told me the project I gave her is really too boring. So I said, okay, what do you want to do? And she said, okay, I want to work on IgA nephropathy. So I asked her, check my literature about IgA nephropathy. So she's going to hope that she'll find all the expert opinion I have. And she came back very quickly within an hour, say, I couldn't find anything among your publication list about IgA nephropathy. So that's the answer. I say, I never researched on IgA nephropathy because it's such a difficult subject. So she pushed me to give her something more interesting. So we speculate that spleen tyrosine kinase being important in the immune system, is there a remote possibilities involved in IgA nephropathy? And we put forward an idea. I would say that it's a speculated hypothesis that the complex of IgA in the patients, somehow they would get stuck to the receptor or surface of kidney cell, in particular mesenteric cells in the kidney. And following that, they would stimulate the spleen tyrosine kinase and to cause release of inflammatory mediators. Now, at that time, we have no evidence whether it's true or not. So we've done some experiments. And some of you have seen that before over the last couple of years. So the first evidence we have is that we, together with Jonathan Barrett and his colleagues in Leicester, we get hold of the serum sample from patients in the freezer, or the serum in the freezer, the patient in his clinic. And Jonathan and his team kindly purified the IgA from them. And then we use it, we've done the easy bit. So we bring it down to London and put it in our cell culture in the laboratory. And then we find that we confirm what other people know is that if you put the patient's IgA and kidney cells in culture, particularly mesenteric cells, they release a whole house of inflammatory mediators. And they are sufficient to cause further kidney damage in a real-life situation. Now, this is not rocket science. People have done that before. All we did is ask someone expertise like in Leicester to help us, and we start the experiment. So the real test is that what if you try to look in the kidney cells of what are the underlying mechanism. And this is a simplified graph of what we did. So we look at the protein expression of spleen tyrosine kinase over time. Now, if you look at time scales very fast, over 30 minutes, then you can see as soon as you put the IgA in the kidney cells, they start to increase the amount of spleen tyrosine kinase, the activated form. For spleen tyrosine kinase to be active, they need to be phosphorylated, which is adding a phosphate group to them. So this is the first piece of evidence we have that spleen tyrosine kinase may have something to do with IgA in patients. And this is work done by the researcher at that time, Minjung Kim. She was the one telling me my research is really boring. So that's how we get started. Now the question is, if we demonstrate the enzyme is there, what is the consequence of blocking that? And can we demonstrate that can reduce inflammation? So we got a collaboration with the pharmaceutical industry at that time. They provide this rare user research tool, which is an inhibitor to spleen tyrosine kinase. And that would block the active site of the enzyme and hopefully to block the inflammation. So we done the experiment at that time. We put this inhibitor in the cell culture of the kidney cells I mentioned to you earlier. We predicted, we don't know the answer, that it would block the production of inflammatory mediators. And that's what we find. So we find here that if we go back to Danny's story about genetics, the next thing below the gene is the gene is affecting production of messenger RNA protein. In here, we look at the messenger RNA of one of the inflammatory mediators. We look at the protein productions. So it's a complex graph, but if I give you a key message is that in the patients, in the presence of IgA from patients, we have production of the messenger RNA protein for this inflammatory mediator up here. As soon as we put increasing amount of this Syk inhibitor in the cell culture media, we can reduce the amount of productions. So this is the first hint that not only we can find this enzyme as a drug target in the kidney cell in culture when we stimulate patient IgA, when we use a pharmacological inhibitor, we can reduce the production of inflammatory mediators. Now, a lot of you probably have a lot of background knowledge about science and inflammation. We all know that one mediator doesn't explain everything. In our body, when we get ill with something, there's a whole family of inflammatory mediators involved. So we screen for a bigger story, and what we find is that the experiment we did, we reduced the production of a full house of cytokines, about 6 different ones together. They got inhibited in the same experiment by this pharmacological inhibitor of spleen tyrosine kinase. Now, there's a problem here. A lot of you know that when you take a medication, you think the medication is doing one thing. They can be doing 5 or 6 different things. They can either be off-target effect because the drug is not selective enough, or it can be a side effect of something that you may or may not desire. So when we got results like I showed you earlier on, we're a bit skeptical. We repeat the experiment about 3 or 4 times. We get the same results every time. But if we just get a gut feeling, it's just too good to be true. It can be something else going on. So we thought we've got to change the approach. We use the same cell culture. This time, we didn't use the inhibitor from the pharmaceutical company. We used a molecular method of silencing the gene expression. And that's what we did in here. So we got silencing RNA that would neutralize any gene expression of spleen tyrosine kinase. And we find out what happened to the cytokine production. And what we find is that we confirm the results we got earlier on. The whole panel of these 6 different inflammatory mediators, they were also silenced by using the molecular method, confirming that pharmacological inhibitor and the molecular method show that spleen tyrosine kinase is a target. Now the question is, all that thing we talk about is the nearest thing to patients is the serum sample from Leicester IgA patients. Other than that, we have no other clue whether it's true or not in real patients. So then that's upon we go back to the kidney biopsy of patients in our renal surfaces. So this is a big archive of sample over time. And one of my PhD students, Steve McAdoo, he done a wonderful job. He sort of started to use immunostaining of this enzyme in the biopsy of patient. All this brown color staining had the spleen tyrosine kinase. And this is negative control with no staining. And what you find is that in real life, every patient is different. And what he found here is very interesting is that when he compared different severity of IgA nephropathy based on the features of a kidney biopsy, of how inflamed they are, the more inflamed the kidney is, the more of the spleen tyrosine kinase expression. The quiet one, the less inflamed one, you can hardly detect any of them. Now that is interesting because we got to face a real world that you cannot expect that every patient respond in the same way. So you got to identify the patient who are likely to benefit by a certain new treatment. So that's our understanding at that time that in future, if this is ever going to be useful treatment, it is the patient who got the more inflamed looking histology of the biopsy. They are the one that we ought to think about as a new treatment. And then what is interesting is that in a wider context is that IgA nephropathy is not the only inflammatory kidney condition involving spleen tyrosine kinase. And we have looked at other conditions like a rare condition called Goodpasture disease, or a medium condition like vasculitis or lupus nephritis. All of them share this mechanism on the renal biopsy there. And what is interesting but also concerning is that when you look at all these patients at the time of biopsy, the one who have more severe renal impairment, like the raised creatinine, they are the one who express a larger amount of this enzyme abnormal in the biopsy. So circumstantially, we have find that in severe disease, you can detect the expression of this inflammatory molecule enzyme, spleen tyrosine kinase. And then they go with severity inflammation. And the question is, where do we go from there? And then quite thankfully, I done something which I didn't think very much at that time. One of the visiting researchers from Australia, he came to visit our lab at that time before we published our first paper. He asked me a very simple question. He said, what are you doing these days? And I know in a few months' time, he's going to see all this data we're publishing. So I told him right away what we're doing, show him the data. And he done something really wonderful. So he go home, back to Australia, and find that very interesting. He tried to repeat every single experiment we haven't published yet. And so over the stretch of 5 or 6 years after that, every paper we publish, he published a parallel paper about 6 months later, confirming every single piece of data we have. And that has been very useful because one of the concerns about medical research is that not only you cannot trust a piece of paper, sometimes I don't even trust my own data. But if you got different scientists or different doctors somewhere in the world confirming your finding, you know that it is worthwhile to carry on. So to summarize where we were at that time a few years ago, we've shown that spleen tyrosine kinase is involved in kidney cell stimulation by patients' IgA. And blocking that using different approach would reduce the inflammation in the laboratory. And what is encouraging is that in patients' biopsy, you can increase the amount of this molecule. So that's when we were very keen to do a clinical trial. And after we back in the industry for a few years, for some unknown reason, they agreed to start the trial. And I was very concerning. They would give me a letter saying that I'll support your trial and help you. And they never gave me the letter because I can't write a grant to get the funding for it. And then they told me the decision is that they would pay for the whole trial, and which is very generous of them. Otherwise, we would not be here today. So what we did is that at that time, we started a proof of concept clinical trial, a phase 2 clinical trial. For those of you involved in clinical trial, you know that there are different stages. Phase 2 is really the intermediate stage, but this one is a good one. We managed to do double-blind randomized control trial. That means that the doctors are blind, they don't know what they're doing. The patients were not told whether they're taking a placebo or not. So we neutralized all those psychological effects of trial management. And we've been supported by Kidney Research UK, and most important among the audience, there's a person sitting here. On the day we designed the trial protocol, he sits among us, and he's a patient. And he tells us clearly what is useful for a patient, what is not useful. So the whole protocol we did at that time was really based on a lot of patients' input. So and then Kidney Research UK continued to support us in terms of patient engagement, which is very useful. And the trial is much harder than we think. At the end, we got to have 20 international centers collaborate together to make it happen. And we started in 2014. And we finished the last patient treatment in March this year. And so where we are now is that we are analyzing the data. But I got 1 or 2 slides to show you the preliminary analysis. But on whole, the trial, what we did is that we really have no confidence because it's the first time this class of medication using any patient with kidney condition. So we play safe. we thought the first thing, don't do any harm to your patients. And so what we did is that we give a short-term trial assessing the safety and how tolerable the medication is, and hopefully get some signal about efficacy. And we only treat the patient for 24 weeks, which is a very short time for a long-term condition. And the key inclusion criteria is that patient got to have a recent biopsy showing IgA nephropathy, features of inflammation, and then they need to still have more than half a gram of protein a day in the urine after their blood pressure is controlled, after they've been stabilized on angiotensin receptor blocker ACE inhibitor, because we don't want to expose the patient who got good kidney function after standard of care treatment to an unknown drug. And the key exclusion criteria is that there are some patients with scar kidney and very low kidney function. At that stage of the development, we thought it's not fair to ask them to join the trial yet. So that's a design of a trial. We try to have 3 groups of patients. At the end, what we achieve is 76 patients internationally, 25 on placebo, 26 on low dose, 25 on high dose. And all of them were given the medication orally. And this is the trial protocol. At that time, the main thing, the message to take home is that initially, there's a lot of frequent visits in the beginning because we want to be sure of the safety of patients. But once they get used to the medication, then the visit becomes less frequent. And the key endpoint we're looking for is whether there's evidence of improvement of proteinuria, any improvement of histology, and the most important, how safe and how well tolerable the medication is. And so this is the distribution of the research center across the world. And UK got 6 hospitals and 20 of them across the world in total. And one of the things we're very pleased is that we managed to engage both North America and the Far East in this study as well. And this is the number of recruitment. You should be very pleased that UK done very well. So we've done 23 patients in UK, 23 in America. So consider America so much bigger. And then we've done really well in recruitment. So the timeline is interesting. We first studied this molecule in 2012 in IgA nephropathy. So now we just finished treating the last patient in the trial. The question is, all of you want to know what the outcome is. I don't know the full answer to that because we have a lot of data we're analyzing at the moment. But what we think is that the patient who have milder disease, we're not sure we can demonstrate the benefit or not. So these are the patients who just got halfway between half a gram of protein in the urine a day after standard of care of treatment. There's a lot of baseline fluctuation of kidney proteinuria. The kidney function is very stable. And it's not easy to be sure whether our drug makes a difference. But now, when you look at the more advanced disease patient, these are the patients got more than a gram proteinuria a day, despite you do everything for them. Then we see something interesting. So there are some improvement on placebo. So you got about 15% reduction of proteinuria. Like all situations, for some strange reason, if you do a clinical trial on many conditions, the placebo group do have some benefit. And then what is interesting is that with the increasing of dose of SYK inhibitor, here you can see low dose, you got further improvement, high dose further improvement. But you notice that for all those professionals around here, I didn't have a p-value here because the statistic is not significant. What we see is just a trend of improvement. And I presume part of it is that when we look at the patient with more advanced disease, we get about 14 to 16 patients per group. So our sample size is too small. And there's no doubt that our treatment duration of 24 weeks is a very short term for a long-term condition. So the safety side is worth reporting. So on the whole, we know about the side effect of the medication. We didn't see any surprises. This drug is known to cause diarrhea and nausea and sometimes headache and hypertension. And high blood pressure is interesting because we knew this drug can cause high blood pressure, so we controlled for that, and patients were well treated. So we managed to neutralize that effect. And then in terms of how well tolerable is, a small number of patients had adverse event, and one patient had a perforated peptic ulcer and died in a Far East center. And then we asked the independent investigator to look at that. They don't think it's related to the medication. But I think it's just fair to let you know that there's one patient gone through that. So in conclusion, I've shown you the data of our preliminary study where we go from kidney cell culture to international clinical trial. But the preliminary data show that patients more advanced proteinuria. They show some benefit to the drug treatment, but the sample size is small. We don't have statistical value to validate that yet. And then we're planning for further analysis and clinical study. And a lot of people help in this study, including, most important, the patient's input, not just being as a trial subject, but actually in the design and also publicity of the trial. Thank you.
Unfortunately, we don't have any time for questions, but
Yeah.
if we see Fred at coffee break, you can ask
Yeah.
him some then. So I now hand over to Phil, who's going to lead the patient panel.
People that are coming on the panel, can you go ahead, please?
Make sure you use the microphone. It's important to the people online.
Hi, everybody. The idea of this session is that this is called a Patient's Day. We've done a tremendous amount of listening to national experts in the field, and this gives an opportunity for patients to talk about things that concern them. And we thought we'd kick it off just by simply having a group of patients, myself included, who are going to spend a couple of minutes each just telling their story, and then throw the meeting open to throw the discussion open to the room. John reminded me to tell you that when we break for coffee after this, the clinicians and the scientists are still going to be here. Okay, so please feel free to buttonhole them out there in the coffee. Questions you want them to answer, please ask. And if there are any questions you want to bring up, if you want to talk about issues about your life and your story, the way in which the disease impacted on you or hasn't impacted on you, then I think we'll do that in the general discussion. Any questions? We're not experts. We're just patients, same as you are. But we can maybe field some of the questions towards some of the experts. My name's Phil Smith. I was diagnosed with IgA nephropathy 8 years ago. And I was picked up in a general call-out from my GP surgery. I was 55 at the time, and he called people together of that age to have a general health check. And I went in. I felt that I was perfectly healthy, although I was working in a very high-stress job. But, I mean, I was okay. And it was found that I got very high blood pressure. And there really seemed to be no reason for that, other than the fact that I was working hard. And that was followed up with a blood test. And that blood test showed that I'd got impaired renal function. And my renal function was down to about 25%. I then was sent to the hospital where John works for a renal biopsy. And the renal biopsy showed that I'd got IgA nephropathy. And that, my condition, my renal function deteriorated at about 1.5% a year, up till last Christmas. And at last Christmas, the consultant who I work with at the hospital, Graham, said, well, you're down to 15% now. You better go on the transplant list. Now, it was quite interesting for me because I'm one of the lucky ones, really. I'm what's called asymptomatic. I had absolutely no symptoms whatsoever through the entire course of this. Even when my renal function dropped to 15%, I was going to the gym 3 times a week. I was having no side effects at all. I was doing the things that they told me about, moderating my diet, and I had a problem with potassium, so I had to eat low potassium stuff all the time. I went on the transplant list at Christmas, and on the 27th of April, I was in bed early in the morning. I got a telephone call, and I picked the phone up, and they said, "Is that Mr. Smith?" I said, "Yeah." And they said, "How are you feeling today?" And I said, "I'm all right. I feel fine." They said, no coughs or colds. I said, who is this? And they said, oh, it's the General Hospital just checking up on you. I said, no coughs, no colds, no chest infections. No, I haven't had any chest infections. Feeling healthy, yeah, eating well, feeling healthy, feeling perfectly okay. And then the phone was passed over to somebody else, and who said, well, and I won't tell you his name, but I'm one of the transplant surgeons at the hospital, and we've got kidney for you if you want to come in. So I said, oh, really? I said, can you tell me a little bit more than that? And he gave me information about the kidney and its function, and although he didn't identify it, a little bit of information about where it had come from. So I packed a bag and went into hospital, and late that night had a kidney transplant. There were a couple of problems I had with it. I had a neckline I'd never had dialysis before, but they needed to take some potassium off, so I had a neckline went in, and that went in a bit wrong. And in fact, the neckline went out of my vein and into my artery, so I had to go to another hospital, have that removed. And I also, a couple of weeks after the transplant, I developed a swelling where my kidney is, which is just here, which they were a bit worried because they thought it might have been a hernia. But it wasn't a hernia, it was a collection of fluid. So I had that fluid taken away, and they've got the fingers crossed because they were hoping it was a type of fluid called serum. And apparently if it's this fluid called serum, when it's been taken away, it stays away. And mine wasn't. It was fluid called lymph, which they really hoped it wasn't. So it came back again. Within a week, it was, I was pregnant again. So I had to go back to hospital, and I had to have some keyhole surgery, and they put a drain in between where the fluid was and where into my peritoneum, and it drained away that way. And since then, it's been fine. The only impact I've had really, as I said earlier when we were talking about fitness, is how you go backwards in your fitness after a transplant, because you do rest and you don't do as much as you did before. And when you start doing proper exercise, it's like you're starting from the beginning again. But it's getting a lot better now. I can swim a lot better, and I can run now, and I can do resistance stuff. So that's me. I will say one interesting thing. You do have interesting contradictions between the work that happens in the hospital and your GP surgery. Because about a year ago, I was called into the GP. I had a message from the GP. Calling people in for a urine sample. I thought, oh, here we go. So I went in and took a urine sample, and a couple of days later, emergency telephone call. Can you come straight into the surgery? I said, yeah, okay. I've been visiting my transplant, my consultant 4 times a year. And every time I went, he sent a detailed letter to my GP surgery. So I went into GP surgery and I saw a locum who said, we're really worried, Mr. Smith. And I said, oh dear, that doesn't sound very good. I said, why is that? She said, you've got protein in your urine. And I said, yeah. She said, well, it's pretty high. And I said, yeah. And she said, well, we need to find out why. I said, well, I've got CKD. I'm a kidney patient. And she said, oh, yeah? And I said, yeah. And she said, how long have you been kidney? So eventually we got it sorted out. So there does seem to be a bit of a disjunction in terms of communication between what happens in hospitals and what happens with your GPs. I know they're very busy. Okay, thank you. Margaret, have you got time to do a quick one?
Yeah, okay. Sorry.
Margaret's got to leave to catch
I'm
train.
really sorry. I've got to go. So I'll just say very quickly then my story. So I was diagnosed about 5 years ago with IgAN, very similar to Phil in the sense that it was picked up just through routine GP's screening, if you like. However, I'm quite sure I had it for many years because I had been treated for high blood pressure for 10, 15 years before that, and it had never been picked up as anything more than that. So I'd just been treated for the blood pressure. So at the time, so I went through loads of tests. It took about a year to get diagnosed with IgAN. Obviously, finished with the biopsy. In my case, 2 biopsies. We just won't go there. It was just... Anyway, it was picked up eventually. But in that year, my EGFR, my kidneys function, dropped from 65 to 35%. So I was told that quite likely within 6 months I'd be on dialysis because my kidney function was failing rapidly. So my consultant and I decided aggressive treatment was the way forward. So even more blood pressure tablets to take blood pressure down, and onto prednisolone and mycophenolate mofetil. Now, I'm quite aware that this is quite a controversial treatment. I have been on this for 5 years, not always at the same level. I started off with 40 milligrams of prednisolone, which you really don't want to be on for very long. I'm now on 5 a day. But mycophenolates, I built up to, well, it's 4 by 500 a day, and I'm still taking those. Now, I have no idea whether that's the reason why the kidney function leveled out or not. But my kidney function at the moment is round about 25%, so I'm not taking any chances here because I haven't got much to lose. But what's interesting is it's dropped round about 10 points in 5 years, which I suppose is about as good as you can expect for a slowly failing kidney. So whatever the reason, and it may just be the aggressive blood pressure treatment, it has improved in the sense that I'm not on dialysis at the moment and I'm not on the transplant list. I'm sure that's just round the corner, but we just keep going. And I think, in fact, that's what I wanted to say, really, because you can't let it dominate your life. You know, I've got a life and I do all I can. Anything I can, I do. And the rest of the time, I just get on with my life. I take my medication, I do some exercise. I must say, felt a bit guilty during the exercise talk, not quite as much as I should. I play golf, so I do lots of walking. And it's interesting, I just go ahead. But it's changed my approach on life quite significantly, actually, because I sort of think, well, if I can do it now, I'm going to do it now, because I don't know if I'll be able to do it next week, next year. And so it's had a major impact on my life. It was never on my list of things to do to be any type of politician. And somehow I found myself being elected as a local councillor and then as a local county councillor. So life is extremely busy. I haven't got time to think about IgAN except at my appointment times, when I'm always very nervous when I have blood tests, etc. I just get on with life and do what I can. It does change your life somehow. You know, I mentioned, I think earlier, excuse me, I get very cold. I'm always cold. And also, it's had a very strange effect on alcohol, or should I say alcohol now has a very strange effect on me to the extent where I just don't drink it. I'm not saying you shouldn't drink it. I just say, for me, the impact is just ridiculous, so it's just not worth it. So I think my body's it's telling me it's got enough to cope with without that, so I can live without that, that's fine. So if you can do it now, do it now. That's my philosophy. And just look after what you can. Thanks.
Hi everyone. My name's Brooke and I'm 20 years old. I was diagnosed with IgA last year in March. The problem first arrived when I was 15 and I'd been to the doctors. I weren't very well, unrelated I think. And she took my blood pressure and it was really high. And she gave me tablets, they always do. And I was really symptomatic for 3 years. I wasn't diagnosed for 3 years after that, till I was 18. I was anemic, low vitamin D I had, and the dipstick tests in my urine were showing blood, even though you couldn't see it. And it was like that for 3 years. The high blood pressure was causing really bad migraines. I had to leave my studies twice. I had to leave my job. And in the end, it was just like life was just nowhere for me. And there was a new GP at the surgery one day, and I said to him one time, I was like, I'm fed up of living like this, to be honest. I just want an answer to everything. And he said that he was going to run a normal blood test for me, which I'd had before. And within 2 days later, he rang me up and he said, your kidney function is at 26%, you have to go to the hospital, which I did. And even then, the hospital didn't think I had a kidney problem. They thought it was my... They thought my high blood pressure tablets were causing my kidney levels to like fluctuate. So I was then sent to a proper kidney hospital, which biopsied me and diagnosed me with IgA. And I assumed that my IgA was quite aggressive, I suppose, because within 4 months I was on dialysis. I'd done 4 months of peritoneal dialysis. I didn't do the overnight one. I used to do 3 drains a day, and it was easy, manageable. I used to take my bags with me if I went out, but I used to get drain pain, so I didn't really like that. And 4 months after, so I'd done 4 months of dialysis, and as soon as I started dialysis, I was put on the transplant list, and 4 months after that, I got a kidney. I actually got the call on Boxing Day last year, and I got the actual operation was on the 27th of December. So that's when I got the kidney, but even then, it's had some ups and downs. A third of the kidney died. They don't know why. They think it got damaged, possibly taken it out of the donor or putting it into me. They don't know what one it is, but it's holding its own, so that's good. And I also contracted CMV from the kidney, so I'm on antibiotics for life to stop that coming back because it gave me an inflamed liver. But at the moment, all is good, and it's definitely improved life a lot. I'm back at college now, whereas I couldn't do that before. And I'm doing A-level biology, chemistry and psychology, which is what I always wanted to do. So it's definitely an improvement. Thank you.
Hello, I'm Richard. Like Phil, I'm from the Leicester area. I was diagnosed with IgA nephropathy in December 2013. A few months before that, I'd had an operation for a hernia. I thought it was healing up all right, but I kept noticing that my ankles were swelling up. And I went to the GP, and he told me to go to the hospital straight away. I had a few blood tests, urine tests. And so my diagnosis was quite quick. From sort of going to hospital, I found out I had IgA nephropathy within a matter of a week or two. When I was diagnosed, I think I had 41% function. A week or so later, we went down to 35, then 30. About a week after that, it was my function dropped away quite quickly. And that year, after I'd had the diagnosis with my symptoms, I was tired, I had very itchy skin, felt cold all of the time. I used to wake up with hot sweats, which was, that's what I experienced. I think the biggest challenge was more psychological. Everything was very uncertain. I didn't know what was going to happen. My mum, who sat over there, said very early on, she said, you can have my kidney. So I was, I'm immensely grateful for that. But still, you still had the whole process of managing the decline of your kidney function, coming to terms with, previously I'd been a very, very fit person. I'd done a lot of running. When I turned up at my running club to run with them, I could barely run for a couple of minutes because of, I think the anemia made me feel very, very tired. So eventually, December 14, I had a transplant from my mum. Compared to Phil and other stories, the transplant was, relatively straightforward. My recovery was, apart from a few problems with my arteries going into my kidney, it was fairly straightforward. So like I said, that was December 14. I got back to work fine. Ever since then, it's been a lot better from a psychological point of view. If things are stable, you tend not to have the same levels of anxiety. Living with kidney transplant is changed my life. I've managed to have a second child, which is I don't think would have happened without the transplant. And I've taken part in the transplant games, which for someone who was previously sporty, has given me an opportunity to sort of carry on the things that I was doing before. And this year I was lucky enough to qualify for the World Transplant Games, which is taking place next year. So that's me.
Well, you've heard what we've had to say, and I'm sure there are lots of people who want to share some information about the things that have happened to them. I don't know, we're not shooting around with a mic for this, are we? It's going to be too complicated if people are just sort of dropping in and out. What do you think?
It's difficult for people on the Facebook page.
Right.
They can't hear anything unless it's on the mic. So unless you repeat the question,
Yep.
that's another way of doing it. So there are two ways to do it. One is we can shove the mic around, the other is you can repeat the question.
Okay. Well, if it's going to be a quick question, I'll repeat it. And if you're going to be talking at length, you can have this.
So if it's going to be a long question, put your hand up.
Yeah, if you. So in terms of a question or sharing a bit of information about themselves or talking about their story, I mean, who'd like to share with the group some of the things that have happened to them? Thank you.
This is actually a question for the panel from a viewer at home. This viewer is really interested on the impact that your diagnosis had on your spirituality. And I just wondered if any of you guys engaged more spiritually or turned away from it, having been diagnosed with the disease.
In terms of me, no, it didn't. It was, I accepted my diagnosis. I felt fairly positive that my consultant told me that although my EGFR had fallen to 25%, I probably had IgAN possibly for decades, and it's taken that long to get that. So I knew that it's probable that this was going to be a slow progression, and it gave me an opportunity to come to terms with it in a reflective way. So in that way, it didn't have any sort of impact upon my view of the world and the universe and all within it.
I'd say my diagnosis did, actually. I suppose before I got ill, I did have a view on spirituality and how the things worked, and then I just found myself really angry or disappointed after diagnosis, because it was, even though I was ill, it was unexpected, and I was just convinced I was going to die, and so it did affect me regarding spirituality. Yeah.
Before I was diagnosed, I was agnostic, atheist, and that's how I remain today. Just the way of the universe, I think.
Anybody like to share any comments from the floor about the way in which their diagnosis impacted upon them? Not necessarily spiritually, but just how you felt about it and how you coped with it, and the impact, maybe the impact on your family as well.
Hello, I'm Brooke's nan, and it does mentally affect on you, because when she goes to an appointment at the hospital, she's in a hell of a state for a couple of days waiting for a phone call. And I'm just wondering, like, anyone else experiences that? Because she's terrible, you know. If anyone else experiences that?
I do. I have me.
Can you have the? Can you wait for the mic? Thank you.
I cyclic from being very, very anxious to just forgetting everything and just thinking I'm absolutely normal. And when the consultant says, "Oh, I'm not going to see you for 6 months," I'm like rejoicing that I can just go back into my world of taking my tablets and forgetting about it. And then as that appointment creeps up, I start getting more and more anxious. And then, you know, when it's every month, or I find myself staying in that sort of, I feel like a sick person, and then it sort of trails off again. But yeah, I do get that anxiety.
Hi. So I was diagnosed at the same time, like when, I was 20, so we're your age now. And what I found was really intense at the beginning, and then you really start to live your life a lot more focused, which is actually it's a bit of a, I wouldn't say gift because it's horrible, but you know what I mean. What I struggle with actually is the dips of infection, and if you get a cold and then you sort of have more symptoms, and it's going from feeling really, really sick and then not feeling sick at all, and other people's reactions. So at work, lots of people come in and they're sick because they don't want to take time off, and I don't know how to deal with that. I don't know how to explain it. I just wanted your opinions on how to explain that to people without seeming really nervy and over the top and like a hypochondriac, I guess, to other people, their perception.
Anybody in the audience like to share a thought about that? How you share information about your condition with others, on the basis that there's a possibility that they could give you an infection. If they sneeze on you, it's not, you know, it's never nice anyway. But if you've got IgAN, it's even worse.
Yeah, I guess people don't know what doesn't affect them. So, you know, being as open and transparent about your condition and how they might affect that is the best policy, really. And don't be afraid to be blunt. It's your health at the end of the day.
Yeah. In the middle first.
Yep. Yeah. Hi there. I just thought I'd share something with the room, which may be useful to one person, who knows. I'm a bit like Phil. I've had quite stable IgA for the last 4 years at about 25%. I live a normal life. I'm quite active. Everything's great. About 4 months ago, I decided to try and lose some weight. So I went on a low-carb diet. For me personally, that was a disaster. My EGFR dropped to 19% and I went into hospital. So just be mindful of that. If you're thinking about a low-carb diet, just watch your EGFR.
Yeah, I was just going to share as well. Similar sort of thing. I found out through just a random, you know, you're of an age, come in, get your tests done, and one of the tests brought up the fact there was blood. And 3 years ago, I finally was diagnosed that I had IgA. In terms of sharing it, I've kept my circle of who knows about it quite tight because I try not to let it sort of rule my life. Yeah. I haven't even told my mother because if she knew about it, it'd be any sniffle or sneeze would be a complete nightmare for her. Or be a nightmare for me, listening to her going, "Oh, what's going on?" and panic. And in terms of, you know, illnesses and being around people, I just came off a cruise last week and somebody described it as a floating petri dish of disease. And I came off a flight afterwards, which was just as bad. Unfortunately, the real world is you never know when you're going to be in contact with people. So I think worrying about it is possibly not to your benefit. I would suggest if somebody does come in and they're extreme, then you keep well away from them. And if you have the possibility of working from home, then that's great. But the reality is you go in the tube, you go to the store, you don't know what you're facing. So take care of yourself, do everything you can for yourself, but don't worry about it.
Yeah, I just wanted to follow on from the contact at work and things like that. So as a transplant patient, I am the only transplant patient that actually works in the office at Kidney Research UK. So it's part of what I do is also kind of educating everybody else in the office about kidney disease. So that people who come to work for us don't necessarily have experience of kidney disease. And so one of the things I do is do lunch and learn sessions where I basically tell them about the kidney patient journey. And we've got it now as part of the induction. Obviously, for Kidney Research UK, it is much more relevant to do. But it could be something that you try to do at work as part of health and well-being. It could be that you ask if you can run a session and help educate people in understanding what it means, because a transplant is a treatment, it's not a cure. And there's so many misconceptions about kidney disease and about transplantation and about what you can and can't do. And by doing something like that, you could really open people's eyes up, and that might help you in the workplace with the kind of germs and things that go around. I mean, I went into work. We have a shared desk policy now at work. So I went into work, put my stuff down next to my colleague Rosie, and she just looked at me and went, "Don't sit next to me today." And she was literally like, she ended up going home because she was so unwell. But she was able to acknowledge that I shouldn't be sitting near her because having a suppressed immune system could catch something. So I think maybe try and see if there's something that you can do at work about kind of communicating and helping people learn more about kidney disease. And the likelihood is there might be someone there who is affected by kidney disease and you don't know about it. Or there might be somebody there who has something going on and doesn't realise yet. So that's always really helpful as well.
Thank you, Sarah. Just to pick up on a point that Sarah had a moment ago, I was in a meeting once when John Feely, who's the former professor of nephrology from Leicester, was talking to an audience. And he said something that I thought was really quite interesting. And I think it's useful to share with others when you're talking about this dimension to your life. He said that anybody who got CKD and renal failure 60 years ago, what happened is you died. Now people don't die anymore in that sort of way, because now what we've got is we've got dialysis and we've got renal replacement therapy, so we've got transplants. He said, but that doesn't mean we've cured kidney disease, because we haven't. What we've got are hold-in positions. And I think the thing that we've heard about today, which is really important, and certainly the work that Kidney Research UK do, is funding research which is based at curing kidney disease. And kidney disease at the moment is not cured, although we've got things that can help people cope with it. Anybody else like to raise anything? I mean, it was quite interesting that people were talking about diet earlier. Does anyone like to comment on the pros and cons of diet that they're? Okay, thank you.
Hello. I had quite severe protein loss at some point. I found that a gluten-free diet actually helps, and my protein loss is reduced. I did have other symptoms that suggested I should go gluten-free, but I've found that I've got a lot fewer kidney symptoms since following gluten-free diet.
Anyone else like to raise anything? Yeah, okay. While the microphone's going there, I'll just say that before I had my transplant, I was on a low potassium diet. There were lots of things I couldn't eat, chocolate and tomatoes. And when I had my transplant, my potassium function went back to normal. And my transplant surgeon said, yeah, you can eat high potassium foods now because your kidney can cope with it. And I thought, right, am I going to have a banana? I hadn't had a banana for 6 years. So I went out shopping and bought some bananas, and I came back and I look, I didn't fancy one. Sorry, someone towards the back?
Yeah, I'd just like to share that, as probably most people know, the journey isn't smooth. I was diagnosed December 15, and it's been a rocky journey since then. 4 months later, I started peritoneal dialysis, which has worked well. We've been away on holiday with 10 days' notice skiing. We packed a bag, 2 suitcases of manual fluids, cleared it with the airline, went away. So
and we go camping. We have electric hookup, but it doesn't have to be the end of things you do. You just have to plan ahead. I view, when I was on manual dialysis, you go out for a day. It's like you've got a baby. You've got to pack your bag and go out.
Yeah.
And but we do try and carry on. Unfortunately, in January, I had a transplant, and I had a blood clot, so it came out 5 days later. And then I had C. diff, so it wasn't nice, but I've got over it. It takes a bit of mental to get your head around it all, but now I'm back on the transplant list, so things can go really wrong, but it takes time. And previously I've had 2 lots of counselling, so if you need help, I'd recommend seeking help. But our view is it's for life. I've got kidney problems for life. Got to make the best of it. It's changed my life. I have afternoon naps, go to bed, but we maintain quite a good family life. So I just like to say, if you do get to dialysis, try and make the best of what you can and do things. That was all.
I mean, you raise an interesting point about it not letting it rule your life, and it's a thing that Margaret said as well, who's had to leave us. That, you know, having IgA in actual fact has galvanised her life really, and she does a whole range of things that she wouldn't necessarily have done before. One of which I know full well, because Margaret's a Facebook friend of mine, is that she travels extensively. She's never at home, other than when she's playing golf. But I wonder if John's here, if you could comment, John, on what you say to people who come to you who've got CKD, who say, what guidance would you give us about travelling? The places we ought to go to, places possibly we ought not to go to, and the things we need to think about.
Thank you for that, Phil.
You can get me back for that.
So I don't think there's any specific IgA nephropathy advice. There is advice relating to what your level of kidney function is. Because the closer you get to needing dialysis, the less reserve your kidneys have of dealing with problems. Be that a bit of diarrhea and vomiting, be that the very hot weather and getting dehydrated. So the advice I give is very much related to the level of kidney function, whatever the cause. And the closer you are to needing dialysis, so if your kidney function is in the low teens or in single figures, then we are very clear about offering advice about where are potentially good places and potentially not so good places to visit. So by and large, travelling around the UK is relatively safe. Europe is okay. We have a lot of patients in Leicester who come from the Indian subcontinent, and we do get concerned travelling that far distance about what would happen if someone's kidney function failed in that part of the world where perhaps there isn't as much easy access to dialysis. And I guess in London, again, there are lots of people from lots of different parts of the world where you have to tailor the advice to the countries that they're going to. But there should be no specific IgA advice. It's very much related to the level of kidney function that you have. The other issue from a transplant point of view are the vaccinations that you're allowed or not allowed to have to go visit certain countries. And that's something you do need to think about in terms of live vaccines and killed vaccines. But my advice to patients is that you let me look after and worry about the IgA nephropathy, and you go on and do whatever you want to do. Because I think that's the absolute key, is everyone in this room needs to be able to work with their kidney doctor, to understand what's going on, to understand as well as possible what's happening in the future. And when you leave that room is to go and live the rest of your life and let us think about how we handle the kidney disease and work with you to try and make things as good as we possibly can. But the important thing is not to let the IgA nephropathy run your life, not to get obsessed with looking at your blood pressure 3 times a day or dipping your urine every day. We will come up with an idea about how frequently you should measure things, what will be an indicator. And to be honest, most of my patients have my telephone number, my office telephone number. I look at the Facebook pages and I contact patients if they're doing something very silly, privately. But you should have a contact number for someone to ask advice. There is nothing worse than having a 6-month review and thinking of something on month 1 and waiting 5 months to ask the question. We need to have, and this is where Tom's talk came about social media. We need to have a much more receptive way of getting advice quickly to stop the worrying, to give very simple, clear advice so that you can get on and live your life. Yeah. And that was one of the ideas behind the Facebook page. And one of the ideas of having these days is that you get contacts with people who you can ask. Because there is nothing worse than sitting at home wondering what the right answer is, going on the internet and finding 20 different answers to the same question, 19 of which will be completely wrong, and not knowing which is which.
Thank you. Well, we're going to wind up in a minute because there's coffee out there and hopefully some of those cookies again. But I wondered if there's anybody who's got a burning desire to make a last point or wants to raise anything before we shoot off for coffee. Yes, last point here then. Thank you.
Just the last point as a mum of a patient.
Yeah.
Reading the lab reports that come, with the level of creatinine or whatever, it's really hard to understand. And I have tried quite hard to sort of look at the level of where we're at. But in fact, if there was some kind of dictionary which was fairly simple about sort of saying, look, this means this, in terms of the kind of reports that you get from the hospital, that would be really good.
Is the patient that you're with on using Patient View?
No.
Right, because I don't know how many people use Patient View here. Yeah, quite a lot of people do. I do all the time. One of the things I like about Patient View is that I always get very twitchy when it comes up to the time for my blood test. And I see my consultant on a Wednesday, and I do my blood test on a Monday, and then I get all my results on Patient View on a Tuesday, so I can read them all before I go and see him. But one of the things on Patient View is if you look at anything, potassium level or creatinine or EGFR or whatever, there's a little key on there as well that opens another page with an explanation of what these numbers mean and what normal would be, and if the numbers are not normal, how abnormal those numbers are, so I find that very useful because a lot of the results I haven't got a clue even what they mean. But if you look them up in the key and it opens the page relating to that, it gives you all the information. So
that's fantastic.
So to get onto Patient View is I would recommend that for everybody actually. It's a great thing.
That's fantastic, and it would be really helpful then if GPs and also consultants could just kind of let people know that where they haven't. Obviously in some cases people have, but
last one this time.
Just one thing from me. Sometimes when you get blood tests taken, and then you phone your GP and just say, what's the result? And tell you, yeah, it's normal. Ask them about the numbers, because quite often I ask them, what's my EGFR?
Yeah.
So 53. Okay. So for them it's normal, but for you might have a completely different meaning.
Yeah.
You have to ask them specific sometimes, because quite often GPs, just for them, just don't really get that.
Well, I think that's an absolutely excellent point. And on that point, we will go and indulge in some caffeine, those of us who wish to. So thank you very much indeed. And thank you, you two. That was brilliant.